it's really my honor and pleasure to be speaking today to this conference and really sharing some of my thoughts about brain health and the development of new therapeutics for Alzheimer's disease. So I'm currently the Vice President of Drug Development at Cerecin, and let me jump right into my presentation. So diseases of the brain really represent a very high economic worldwide burden. And it's predicted in 10 years' time, this burden will be higher than that of cancer, diabetes, and respiratory diseases combined. And if we look at the graph on the right-hand side, about 80% of patients with CNS disorders are either unserved or underserved, which really represents a very high unmet medical need. So there's really a demand for new therapeutics in brain health and in particular Alzheimer's disease. If we focus on Alzheimer's disease as a major disease of the brain, age is really one of the more significant risk factors. And with the aging global population, you can see that there's going to be a very huge explosion of cases, not only in North America, South America, and Europe, but also in Asia. In particular, the population bases in China and India really represent an explosion of new Alzheimer's disease cases in that geographical region. So in this context, Alzheimer's disease really represents a worldwide problem, a very global problem. And previously in talks, I would say it was a pandemic, but I think that's probably not a word I want to use in the context today. But if we look at Alzheimer's disease is a progressive disease. It gets worse as a function of time. And currently there are no cures. As I mentioned previously, age is a significant risk factor, but there are also genetic risk factors, such as the ApoE gene. The two predominant pathologies associated with Alzheimer's disease are beta amyloid plaques and neurofibrillary tangles. All of the current approved therapies today treat only the symptoms of Alzheimer's disease and not the underlying pathology. However, there are many therapeutic agents currently in clinical development and really focusing on the two major pathologies of Alzheimer's disease. That is the amyloid plaque and the neurofibrillary tangles. In fact, just recently, earlier this year, Biogen has submitted their drug registration package to the FDA for aducanumab, which is an antibody to the A-beta pathological protein. And in just 3 short weeks, the FDA advisory committee for this therapeutic is scheduled for November 6th. So these are exciting times, and hopefully we'll be having new therapeutic approaches. Now, the success rate of developing new drugs in Alzheimer's is very poor. As you can see, in a 10-year period from 2002 to 2012, only 0.4% success rate, compared to 19% success rate for oncology therapeutics. In fact, if we even just take the 10 years preceding 2017, there were no new drugs approved for Alzheimer's disease. In fact, the last approved drug was memantine back in 2003. But this is not all doom and gloom. There are actually a number of new targets and mechanisms being pursued. And the hope is that this mechanistic diversity in approaching and attacking Alzheimer's disease will hopefully yield some positive results. What I like about this graph here is it shows very nicely the longitudinal changes that occur in the course of Alzheimer's disease. The dotted vertical line represent the point in time in which a patient will go in and get a diagnosis of Alzheimer's disease by a clinician. What you can see is that there are many physiological and pathological changes that are happening years prior to the diagnosis of Alzheimer's disease. In some cases, these physiological changes are occurring 30 years prior to the diagnosis. If we look at the two classical pathologies of Alzheimer's disease, namely A-beta deposition and tau tangles, which can be seen in the cerebral spinal fluid of a patient, those pathologies are increasing or starting to increase at least 10 and definitely 20 years prior to the actual diagnosis. And the change in these pathologies really parallel what you see in terms of the changes in the clinical manifestation, which in this graph is denoted by CDR sum of the boxes, a clinical outcome measure. Perhaps less appreciated are some of the other changes that are going on in the brain of an Alzheimer's disease patient. What I find really interesting is if you look at glucose metabolism, so this is the ability of the brain to use an energy substrate, that this decreases in parallel with the increase in the appearance of the hallmark pathologies. So the brain starts becoming less able to use glucose, and at the same time we see the pathologies start to increase. Now, whether it's a causal effect or a manifestation of the pathology, we don't quite know right now, but we do know that the observation is that the brain fails to be able to use glucose as an energy substrate. If we ask the question, why might this change in glucose metabolism be important and significant? And I just want to turn your attention to let's look at some facts and figures about the brain. If we look at the brain represents about 2% of the total body weight. So our brains are about 2% of our body weight. Our brains use about 120 to 130 grams of glucose per day. That's how much sugar it uses in order to fund its metabolism and to produce energy. In fact, about a quarter of all the blood glucose that we use in a day is actually dedicated to the brain. So the brain is a very highly metabolic organ. To see that, the brain also receives about 15% of our cardiac output. So all of the blood that our hearts are pumping, about 15% really goes to feed the brain. And if you look at oxygen consumption, about 20% of our total body oxygen consumption is really used by the brain. So the brain is a very central organ in metabolism and the utilization of energy. Now we can visualize what is going on in the brain by using a technique called FDG PET. So fluorodeoxyglucose. It looks at glucose uptake, and it's a measure of glucose utilization. What you can see in the figure on the right-hand side is that in a normal brain, the brain lights up very brightly, which means that there's a lot of FDG PET signal, meaning that there's uptake of glucose in the brain of a normal human being. But on the bottom pictures, you can see the brain imaged of an Alzheimer's disease patient. And you can see the FDG PET signal is much more blunted and muted. And that is a manifestation that the Alzheimer's disease brain is not able to use glucose as effectively as a healthy, normal individual.
And Bruce, there is an attendee who's wondering, what is the normal projection of all these biomarkers in aging adults?
So a lot of these biomarkers are changing during the course of age. So glucose utilization decreases. In fact, if you hold that question just one second, in a couple of slides, I'll actually show what glucose utilization looks like as a function of age. What's really exciting, though, and what is, I think, insightful in terms of what's going on in the brain, is that the glucose hypermetabolism, the inability to use glucose, is very regionally specific. So it's not like the whole brain isn't able to use glucose, but there are very specific regions in the brain, most particularly the posterior cingulate, the parietal lobe, the temporal lobe, and the prefrontal cortices. And what is really interesting is that those regions of the brain also map very nicely to the regions of the brain that are affected by the pathology of Alzheimer's disease. And it's also known that this gene that I mentioned earlier, the ApoE4 gene, also seems to be involved with this lack of glucose utilization. So there is a connection there. Now, glucose hypometabolism maps also nicely not only to the regions of the brain, but also to cognitive impairment. So what we're seeing in this figure here is here are serial FDG PET scans of the same individual over a time course of years. In the very top part, you can see at the age of 69, the MMSE score was 29, meaning this person was cognitively normal. And if you look at their PET images, they're very nice and bright. So their brains are utilizing glucose very effectively. In just 2 short years, this individual started to get an MMSE score around 24, which puts them into what we would call the mild cognitively impaired population. And you can see that the PET images are not as bright as they were when that individual was 69 and an MMSE score of 29. What's really telling is just 3 years later, at the age of 74, this person had a cognitive score of 23, putting them in the mild Alzheimer's disease or mild cognitively impaired population. And you can see in their FDG PET scan that there really is a decrease in their brain's ability to utilize glucose. So this maps very nicely. Now, to answer the question that was posed previously, let me turn on to the next slide here, where if we lead this whole idea of glucose hypermetabolism in the brain really leads to a concept known as the brain energy gap. And we can see that on the left-hand figure, that in healthy adult individuals, the cerebral metabolic rate, or the rate at which the brain is actually metabolizing or generating energy, is very, very high. You can see that it's somewhere around 33 in terms of the CMR score. And if we look at the blue bar versus the orange bar, blue bar represents the amount of energy that is derived from glucose, and the orange part of the bar represents how much energy is generated by ketone bodies, and I'll get into that in a little bit later. As I mentioned, as we age, our CMR rate really starts to decrease. And so if you look at the second bar from the left, a cognitively normal, healthy older adult, now that their CMR, their brain metabolism rate, is actually decreasing a bit compared to a young, healthy adult. As we go down the path to people with mild cognitive impairment and people with Alzheimer's disease, you can see that there becomes this energy gap compared to where that individual is to what a healthy young adult would be. And the other thing I want to point out here is if you look at the orange part of the bar, it doesn't really change very much, either as a function of age or a function of pathology or disease. So the ability of the brain to utilize ketone bodies doesn't seem to be changing, and it really is a change in the brain's ability to use glucose. We then focus our attention on the right-hand side, and if we look at the consequences of the brain hypermetabolism, for Alzheimer's disease, it does seem to be specific for glucose. The brain can't use glucose as an energy substrate. What this does is it leads to the brain energy gap, which then in turn results in a cascade of events leading to various neuropathologies. So a lot of the pathologies that we see in these brain diseases, these neurodegenerative diseases, can be mapped back to a lack of the brain's ability to generate energy. This forms a very vicious cycle that the pathology itself also contributes to the lack of the brain's ability to use energy, causes more pathology, and we go down and with this snowballing effect of really the degeneration of the brain to the part of a disease condition. Now, this glucose hypometabolism has recently gained quite a bit of interest. In fact, just earlier this summer, there was a very nice Nature Review Drug Discovery article published, which really focused on the role of glucose or brain hypermetabolism and the consequence to a number of neurodegenerative diseases. And what's interesting is this hypermetabolism, the inability to use glucose, doesn't just affect neurons or the astrocytes, but it actually affects all of the cells in the brain. So here on the left-hand figure shows the types of changes that can occur in a neuron. But I also want to point your attention that there are also changes in microglia. And microglia are the immune cells within the brain, and they contribute to a process known as neuroinflammation. And neuroinflammation is becoming a very important kind of consequence or manifestation of neurodegenerative diseases. And it probably plays a very important role in this vicious circle, this cascade of events of damage and then further, further causing further damage. So we have to understand that the energy deficit is affecting all of the different cell types within the brain, and this does lead to detrimental consequences. So if we look specifically at neurons of the brain is very energetic, and so the neurons in the brain really use this a lot of energy for action potential. So this is the way that neurons communicate with other neurons. Ion current maintenance, so their general health, neurotransmitter secretion, transport within the cell in terms of its ability to repair itself, which is also reflected in a process known as autophagy, the ability of a cell to clear out misfolded proteins. And don't forget that both of the pathologies of Alzheimer's disease are misfolded proteins, both A-beta as well as tau. So with this context, let me switch over to tell you a little bit about Cerecin's program, our lead program called Tricaprilin. So our program really is trying to fill this energy gap. So I mentioned that in the course of aging, in the course of Alzheimer's disease, the brain is no longer able to use glucose, and so it becomes energy starved. Well, I also showed in that figure that ketone bodies can produce energy for the brain. And ketone bodies come in three major flavors: beta-hydroxybutyrate, acetoacetate, and acetone. These are all generated from fatty acids, and they're all generated predominantly in the liver. So the liver can take fatty acids and then convert them into ketone bodies. And our hypothesis is that these ketone bodies can then traverse into the brain very freely. And we know that ketone bodies can cross the blood-brain barrier, and they can be used as energy substrates for the brain. This is all done with the citric acid cycle. So you can see on the left-hand side that normally glucose through 11 steps can go into the citric acid cycle, where then it produces ATP, the energy substrate for all cells. Now, ketone bodies can bypass some of the steps of glucose metabolism, and they can be converted directly into a substrate that can be metabolized or oxidized in the citric acid cycle to form ATP. So the way that we're thinking about this is, if you look at the figure on the right-hand side, I've shown you this before, that as the brain ages, or in the consequence of pathology, that becomes this energy deficit. And so our working hypothesis is that ketone bodies can possibly refill this energy gap and actually provide energy for the brain to prevent this vicious cycle of neurodegeneration. So we conducted a few years ago a phase 2b study, which was a multi-center double-blind placebo-controlled study. We enrolled 152 patients with mild to moderate Alzheimer's disease, and we treated them for 90 days with either Tricaprilin, our drug that produces ketone bodies, or placebo. The study design was that the patients could be on their standard of care therapy, and at the time it was acetylcholinesterase inhibitors. And what we found is that in our trial, about 75% of the patients were on cholinesterase inhibitors. They were given 20 grams of Tricaprilin once daily compared to placebo for 3 months, and then there was a 2-week washout period. The study was conducted in the United States and performed at 23 clinical trial sites. And we genotyped the patients coming into the study because, as I mentioned, ApoE4 is a gene that is a risk factor for Alzheimer's disease. Here's real quickly the demographics. The groups of treated as well as the placebo group were very evenly balanced, and we saw populations which you typically see in an Alzheimer's disease type of clinical trial. And here are the results. The primary endpoint for this trial was the ADAS-Cog. This is a cognitive scale that's commonly used in Alzheimer's trials. And what was very interesting is as we looked at the placebo group, the cognitive performance of the placebo group, even over this 3-month period, declined when you look at the change in ADAS-Cog score as a function of treatment period. And that's seen in the graph on the left in the darker bars, either the solid gray or the dotted gray bars. So there was a decrease in their cognitive performance. And quite interestingly, the subjects that were treated with tricaprolin actually had a cognitive improvement as seen by a mean change from baseline in the ADAS-Cog scores. In fact, at day 45, we saw almost a 5-point increase in the ADAS-Cog. And after 3 months of treatment, there was about a 3.4% change from baseline in the ADAS-Cog score. And statistically, they were both different from placebo with p-values of less than 0.05. What you can see that in the washout period, when we took away the drug, the improvement in the treated group declined a little bit, meaning that there was some washout there. Now, this is the data for the ApoE4 non-carriers. So these are people without that risk factor. And quite interestingly, is if when we looked at the data that came out of the patients that had the ApoE4 positive allele, that these patients did not show this type of improvement in the ADAS-Cog score. This had us scratching our head a little bit. We didn't really understand, why would genotype affect the ability of the patient to respond to tricaprolin. We did a number of PK studies to show that the genotype doesn't affect the ability of the patient to create ketone bodies. They're exactly equivalent. But in a post hoc analysis of the data, what we did find is that there was a very significant correlation between the ketone body levels and the change in this cognitive measure. So this is telling us that the ApoE4 non-carriers seem to be able to respond or to utilize the ketone bodies to result in a change in cognitive performance, where you can see on the right-hand side that the ApoE4 positive carriers were not able to have that type of response or correlation. To follow up this observation, there was a very nice study done at UCLA. This was an investigator-initiated trial where they actually looked at oxygen utilization PET. So they used oxygen PET to really look at regions of the brain that were very metabolic, that were oxidizing, that were energetic. So a measure of energy utilization. And what the UCLA study was able to show is that indeed, when these patients were taking our tricaprolin supplement, that the brains of the ApoE4 positive patients that can be seen on the right-hand side of this figure, they are not able to use or utilize the ketone bodies that are actually produced from tricaprolin, where the ApoE4 negative patients are able to very effectively use this as an energy substrate. This is a very interesting observation, and we're actively trying to dissect out the molecular mechanism for that, but it is a very interesting observation. So let me conclude by thanking you for attending today's session and the Precision Clinical Trials meeting. Very exciting time for Alzheimer's disease, and we're really hopeful that this diversity of approaches of attacking this complex disease will hopefully yield some beneficial therapeutics coming out there in the future. Thank you again.
Thank you so much, Bruce. And we do have a question for you over here. Was the study powered to look at the ApoE4 carrier separately?
Yes, the study was prospectively looking at the difference between ApoE4 positive and ApoE4 negative. But I do remind you that this was a phase 2 study it was more exploratory and definitely not pivotal and definitive.
I think in terms of your hypothesis, you mentioned a couple of times that it's not clear what's cause and effect. Do you think in the overall pathophysiology with regard to the role of beta amyloid and tau, do you think that the glucose metabolism deficit is a mark of dying neurons or missing neurons?
You know, it's interesting because I think that the decline in glucose metabolism by the brain is just a consequence of aging. And then what happens is when you have pathological insults, it really starts this snowball effect. And so I don't think it's dying neurons, but the neurons become, and some people have even coined this term, type 3 diabetes, that the neurons become insulin resistant, so they're unable to use glucose. And maybe this is a consequence of diet, lifestyle, or just even a consequence of aging. We really don't know. All we know is the observation that the brain's ability to use glucose goes down with age, and it's actually accelerated when you have these pathological insults. Not only in Alzheimer's disease, but you also see it in other neurodegenerative diseases.
Interesting. What was the adverse event profile in your treated patients?
For tricaprolin, our predominant adverse events are really gastrointestinal. The dose of 20 grams once daily really causes a pretty high burden on the GI tract. And so we see mostly nausea and a feeling of bloating. We don't see any changes in any of the other clinical chemistry measures that we've looked at.
And my last question is, have you or others looked at differences in the glucose utilization between keto diets versus carbohydrate-rich diets?
Yes, you know, that's a very active area of research. In fact, Stephen Cunnane, who was the first author of that Nature Review Drug Discovery article, has been really extensively looking at ketogenic diets and brain utilization of ketone bodies, as well as using ketone body supplements. So things like tricaprolin, our molecule, but there are also ketone body esters out there and ketone salts that are out there, more on the dietary supplement side.
And that reminds me, one more question. Do you look at this as being a nutraceutical or a therapeutic ethical drug kind of pathway for approval?
Excellent question. About 6 years ago, Cerecin really took the path that in order to get as many people access to this product, that we were going to take the pharmaceutical approach. And so tricaprolin is a pharmaceutical product. We're generating it in GMP fashion, and we're conducting all of our clinical trials under ICH GCP. So we really want to do this the right way and get this to the market, get patients access to this drug really globally and worldwide.
Great, fascinating, thank you.
Thank you.
There's any other questions? Oh, there is one from Molly Sarkar, and she's asking if the ability to generate ketones via the liver is the same between APOE plus and background. What is your hypothesis why positive genotype is not able to utilize ketones in the brain? What percentage of AD is negative genotype? So it's a long
Excellent question. Something that we've been discussing internally for quite a number of years. We've done PK studies to show that it's not the ability of the liver to generate ketone bodies. So if we were to give tricaprolin to a 4 positive versus a 4 negative patient, their ability to generate ketone bodies is the same. We don't see a difference. That's point along with the UCLA results is it's the brain ability to use those ketone bodies, which seems to be different in the 4 positive versus the 4 negative patient. We still don't know molecularly what's different, but it's probably something to do with the ability to utilize it, maybe the metabolism, but we haven't really figured out exactly the precise defect that you see in the 4 positives.
If there's anybody else with any other questions for Mr. Bruce, do let us know. He's a well-known speaker. He speaks at our summits. He was supposed to speak at the hotel today, but unfortunately, due to the COVID situation, it is virtual. But we do plan to have Bruce back at our in-person summits, hopefully by Q3, Q4 2021. Fingers crossed, exactly. And there is one more question. Molly's asking, what percentage is AD negative or NEG?
So this is interesting. If you look at sort of the Western populations, the ApoE4 positive population tends to be more ApoE4 positive. So it's about a 60-40 split of A4 positives versus negative. And what's quite interesting is if you look at the Asian populations, that ratio is actually flipped exactly opposite. So if you take a Chinese Alzheimer's disease population, about 60% of those individuals are ApoE4 negative, and only about 40% of them are ApoE4 positive. So there are some geographical differences in terms of that gene distribution.
Once again, Bruce, thank you for your presentation, and we'll stay in touch.
Thank you. Thank you.